Hello, it's been a while since I last saw you. So yeah, I'm just about finishing my reservist and I have some time to prepare some lessons for you. Alright, so today we are going to talk about practical skills. This is something that you will be tested on in your WA1 and of course in your practical exams. For the pure bio, right, you will be having a paper, 40 marks paper that in your O levels for your practical, and for the combined is about 25 marks. Can't really actually remember I think it's about 25 marks. Okay, so anyway, we all need to know these skills. Okay, so let's open up your workbook, the practical workbook I mean, to the first few pages and we look through the content pages. Okay, over here. Okay, the skills build up. Okay, so what do we need for science practicals? We need to learn how to use apparatus and materials appropriately and safely. Okay, I know that we haven't had that much experience, even in Set 1, Set 2, going to the lab and all that, but don't worry, alright? As long as you do everything safely and you think about what you're doing, I'm pretty sure that it should be quite self-explanatory. Okay, then follow instructions in an organized and safe manner. So this part really requires people to be able to visualize the instructions in their mind and follow it correctly and not do it the wrong way. Okay, then make and record observations in a suitable format. So what are different formats? There can be table format, there can be graph formats, or chart formats. And take precise measurements, analyze results and come to an appropriate conclusion, plan an experiment, evaluate the procedures stated in the plan. Okay, so these are all the practical skills that you will need to have at the end of your Sec 4 life. Not at the end of life, but at the end of your Sec 4. And so yeah, let's get straight to it. Now this part here, basic safety procedures, you can just pause the video right now, read it yourself. Okay, these are all things that you should already know. Yeah, so I won't talk about it. Same for this. These are, just run through quickly. This is an explosive symbol, flammable symbol, oxidizing symbol. Difference between these two is that there's an O here, so O for oxidizing. Compressed gas, corrosive, this is what you might be handling, some acids or some alkali. Acute toxicity, moderate hazard or irritant. Serious health hazard or carcinogen, this is a symbol for cancer. And last one, hazardous to the aquatic or just the general environment. Okay, so what are the good practices and habits when carrying out experiments? Okay, it's important to develop good habits for practical skills. What should we do? Start by reading thoroughly before the experiment. Okay, so when you receive your practical paper, don't rush to start doing everything. Just read through and try to visualize what you have to do in your head first. Okay, if you're bad at like visualizing, then you can maybe hold certain things and you try to like imagine that you're doing it, but you don't do it. Okay, so you just like touch the thing, then you just imagine that you're doing it. Okay, but then you read everything and then you finish. Okay, so read carefully the aims, introductions, and procedure. Highlight the important points to prevent missing crucial steps. Okay, so this is something I want to enforce for all my students. Get a highlighter for your practical, highlight the keywords. Okay, yes, it may feel very rushed, it may feel like you're wasting time, but if you do this consistently, the more you do it, the faster you become. Okay, so at the start, yes, maybe it becomes a bit slow, but as you train yourself, you are able to catch up and speed up already. Okay, then create a mental image of how the steps will follow each other. Okay, I know some people can't really like create mental images properly. I don't know why, but this is something that you also can train. Okay, so the more practical exams that you do, the better you become at this. Okay, then organize your workbench, right? Put away apparatus or chemicals that will not be used. Label the test tubes to avoid confusion as current experiment. In experiments that use enzymes or alcohol, put the enzyme solution and alcohol far away from any heat source. Okay, so why? Because enzymes can be denatured by the heat, right? Especially if it's a high temperature, and alcohol is flammable. So if it touches the fire, the alcohol will catch fire and you'll burn up the whole lab. Okay, then complete your experimental report before washing up. Do not dismantle the experimental setups or throw away any samples before completing your report. Quickly redo parts of the experiment where you find some discrepancy in the result. Okay, so when you're having an exam, you should only wash up when you're very, very sure that you have finished everything. Okay, don't you do halfway, then you want to wash, then after that you realize that you did wrongly, and then you need to reset up the whole thing, then there'll be a waste of time. Okay, and pay attention to these tasks at the end of every practical session, such as turn off the water and gas, return all materials to their proper places, clean your work area, wash hands thoroughly. Okay, so it's all the good habits that you should have. Alright, let's talk about this taking measurements. Okay, this is a very important part here. There are a few apparatus that we can use, right? And we must use them correctly. Let's talk about temperature. You may be given a standard alcohol thermometer, okay, not a mercury thermometer, but what is inside the thermometer is likely to be alcohol. Alcohol is able to, its color of course is red color, and is able to expand and contract based on the temperature. So the bulb of the thermometer must be fully submerged in the liquid when taking temperature readings over here. It should not be touching the bottom or sides of the container. Okay, so try not to let it drop or try not to let it slide to the sides. You just hold it in the middle and then wait for a while for the thermometer to change. One more thing, try to avoid using this thing as your stirrer. Okay, I know sometimes you may be tempted, and since it's already in, right, you want to stir the thing. Because this thing is a bit more fragile as compared to your proper glass rod stirrer, the bulb might break, and then if it breaks, the alcohol will leak out, and then you lose a whole thermometer. Okay, it's not that expensive, but you know, it's still like a waste of resources. Okay, now let's talk about this. Holding the thermometer upright and take the reading at eye level to avoid parallax error. Okay, so this is the same for your thermometer, this will be the same for your measuring cylinders, and even the thing about stringers. Later I'll talk about stringers. Okay, lastly, read off the scale at the bottom of the meniscus when using an alcohol thermometer. Okay, so if let's say this is the reading of the thermometer, okay, so let's say it's 30 degrees Celsius, 40 degrees Celsius, what is the reading now? You have to look at the bottom line. Okay, so bring your eye down to the level of the thermometer, look at it with the bottom of the meniscus. A meniscus is a curved surface of the liquid, and you must count the bottom. So 35, 36, and then you see it's dotted in between, right? Okay, so that's actually not 37, it's 36.5. Okay, so this is what we also call the precision of the instrument. For this instrument, the thermometer, you're only able to read up to the 0.5s. You cannot go any like 36.6 or 36.7. That is a bit too you can't tell that. Okay, you wouldn't know. But you can tell that this is 36.5. Sorry, not 36. 30. Yeah, sorry, 36.5. What am I talking about? Okay, anyway. Now let's talk about stopwatch. Okay, so stopwatch is what I use to count time. Usually you need to measure things for a set amount of time, maybe 30 seconds, you count the number of bubbles, or 1 minute you count something, or 3 minutes for your food test, that kind of stuff. Okay, so how do you read a stopwatch? It's quite self-explanatory. I'm sure that all of you, by pressing it, you'll be able to figure out. But basically, this is the important button here on the right. You press start, then when it hits 30 seconds, you press stop. Okay, and then you want to get it back to 0, you press this button on the left called reset. So what must we be aware of? Firstly, seconds, right? There are 60 seconds in 1 minute, but there are 60 minutes in 1 hour. Okay, so they're quite obvious, everyone knows that. But there's something that you might not know about this thing called milliseconds. Okay, so what is a millisecond? A millisecond is 1/100th of a second. That means that there are 100 milliseconds in 1 second. Okay, so it's a bit funny. 100 milliseconds is 1 second, then after that 60, then 60. So the scale a little bit strange, but yeah, that's just how it is. Okay, so this, most likely the last part over here is counting the milliseconds. You can, most of the time you can ignore that, just focus on the seconds and on the minutes. And some stopwatch will even have the hour. Okay, so I think this one, depending on the model, right? Some models have milliseconds, some models do not have milliseconds. The one in our school has a millisecond counter. Alright, so let's talk about volume. Volume is taken using a measuring cylinder to measure volume. Of course, you must choose the appropriate measuring cylinder. You cannot say that I want to measure only 2 cm cube of the substance and then use a 100 cm cube measuring cylinder. That will be way inappropriate. Okay, or you want to measure 2 liters of, or not 2 liters, 2 meter cube of the liquid, and then you only use a 10 cm cube measuring cylinder. Then that's too small already. Okay, so and the same thing, alright, for measuring cylinder, take the reading at eye level to avoid parallax error. Okay, so just most of the time you should put it on the flat bench, right? Put your measuring cylinder on a flat bench, then you bring your eye down, and then you look at it. Okay, so you don't like lift it up to your eye and you see. Okay, because if you lift it up, you might be holding it sideways or upside down or something. Okay, then the thing spill out. Anyway, you look at the bottom of the meniscus and read off the scale. Okay, so if this is 40 cm cubed, then you see the top one 50. Okay, the middle line, this one will be 45, so below it will be 44. Okay, for this you can put, you can at max you only can tell whether it's 44.5 or 45. Okay, so here is not 45.5, it is 44. Okay, that means that the precision of this instrument is only up to the 0.5s. Okay, what else can I say about this? Okay, some liquids, right, they form a meniscus. For example, water. Other liquids may not form a meniscus. If they do not form a meniscus, then you just read the bottom. Okay, this one is very important. Please highlight this one, the stringe. Okay, because in exam questions, they love to test how to read the stringe properly, or what are the errors people make when they read a stringe. Okay, so firstly, the stringe is a bit different from the measuring cylinder in the sense that you are sucking it up. Okay, but there's some similarities also. First similarity, you must take a reading at eye level to avoid parallax error. Okay, so you need to bring the stringe up. Okay, in this case, you have to bring the stringe up to your face, not go down to look at the stringe. Okay, so you bring the stringe up, make sure your stringe is straight in front of you. Then you look at it at eye level. What should you be looking at? Okay, because there's many parts of the stringe. Should you be looking at this area? Should you be looking at the bottom area? Okay, no. What you should look at is the top ring. Okay, the top ring is this line over here, not the curved line, but the straight line over here. Okay, so do not read the bottom ring or the raised section of the plunger. So you're reading this part. So how can you tell? Let's say, for example, this is 1, 2, 3, 4, 5, 6, 7, and 8. So that will be 8 something. Okay, this one never put the unit, but it should be 8 something. Okay, so be aware of this. Stringe is actually very common exam question in the O levels. Okay, light microscope. Okay, most likely you have already done this experiment. Okay, with me. So just read through. Just remember the basics. Okay, your light is produced by a light source, goes up. Your diaphragm controls the amount of light. Then it goes through the objective lens. There are 3 objective lenses: the 4 times, 10 times, and 40 times. Those are then multiplied again with the eyepiece lens, which also is a 10 times lens, which means that the most magnified you guys can get will be 10, 40 together will be 400 times magnification. Okay, there is another lens which is a 100 times objective lens. You can go up to 1000, but in schools we don't have that. Okay, the rest, such as how to hold, how to adjust, you just have to figure it out. Okay, I mean, we already tried it, so you just need to remember. It's very unlikely that the O levels test you on how to use the microscope. Why? Okay, the simple fact is that it's too expensive to buy for everybody. So imagine if you have a full class of pure bio students, 40 of them, you can't possibly have the whole class using the microscope. So you think about the logistics. Okay, sometimes you have to be smart also. Think about what kind of exam questions can be set based on logistics also. Some things cannot be done because it's too expensive. Okay, so how to hold a microscope? Hold it upright with both arms. One hand should hold the arm of the microscope, other hand support the bed. So it's like you hold this part and then the bottom part, like carrying a baby. Okay, then when you use the microscope, wipe the eyepiece and objective lens gently with a special lens cleaning wipes to ensure they are clean. Do not use the paper towels or any other cloth, because those are rough. They'll scratch the lens, and once you scratch, you will see all the lines inside, and that's very bad. Okay, after using the microscope, rotate the nose piece back to the lowest objective lens. Adjust the stage to the lowest position. Okay, this one we all learned before already. Okay, so this part, I'll let you read it yourself. Okay, like I said, we have tried it. So honestly, you just need to keep trying the microscope. You'll learn how to use it already. Okay, now here is what I want to talk about. These are what we call as the precision of the instruments. Okay, and this is something that you must be aware of. So this table shows some commonly used apparatus in the laboratory and the corresponding accuracy values for each apparatus. Okay, so note, the smallest division of each equipment is dependent on the instrument. The precision of the instrument refers to repeatable measurements and can be obtained from the equipment when it's used to measure a fixed object or quantity. Alright, okay, so let's look at this. The first one is what I call as a mass balance. Okay, I prefer to call it mass balance rather than weighing balance, because the correct unit for mass is in kilograms or grams or milligrams. Okay, so that whereas weight is a force, the unit for weight is actually newtons in physics. Okay, so that's why we should call this as a mass balance. Okay, what is the smallest division? Okay, of course it depends on the model of this mass balance. Okay, it can go up to 0.01 grams, which means that the precision is 0.01 grams, and an example would be 1.65 grams. Okay, or if it's a bigger version, it can only go until the smallest division is only 0.1 grams. Okay, which means that precision is 0.1 grams, and therefore you can only record up to 7.6. Okay, so sometimes exam question will ask you like, oh, state a source of error. Like what could be a source of error in the experiment? Then you can say something like, oh, maybe the mass balance that was provided did not have sufficient precision. The instrument precision was not enough, and therefore we could not get an accurate result, that kind of thing. Okay, let's also talk about this one, lab thermometer. Okay, the unit is in degrees Celsius. Okay, technically, because if you're in Singapore, use degrees Celsius. US, they will use Fahrenheit. There's a common scientific unit known as the SI unit. The unit for temperature is actually Kelvin, but for convenience sake in Singapore, we use degrees Celsius. Okay, so the smallest division actually is 1 degree Celsius. That means that between each line is 1 degree. But the equipment precision is 0.5. Why is that the case? Like we saw just now in the previous picture, you can actually see that there is a difference. Okay, as long as it's in between the lines, right? Like it's in between that line, then you can say that it's 0.5. Okay, so like you can say 37.5 or 38.5. But you can't say 37.4 or 37.6. Okay, because the precision is only 0.5. So it must be in intervals of 0.5s. Okay, ruler. A ruler units cm. Alright, the precision is 0.1 cm. Okay, which is 1 mm. Okay, remember 10 mm is 1 cm. Okay, please don't mess that up. Okay, so here, like we said, the stopwatch. The stopwatch, depending on the model, it can be either measuring in milliseconds, which would be 0.01 seconds, or it can be in tenths of seconds. So like 10th, that means it's 0.1 seconds. So here, is the examples given here. What else can I say about that? Okay, 100 cm cube measuring cylinder. You have other types of measuring cylinder. You have a 10 cm cube measuring cylinder. You have a 1 meter cube, which is 1 liter measuring cylinder, and many others. Okay, but this is the most common. So the precision for this is again 0.5 cm. Okay, how do you know? You read in between the lines. So for example, you can get a value of 50.5 cm cube. Okay, by the way, please be aware of how to measure the difference in a measuring cylinder. Like if let's say I have 50 cm cube in my measuring cylinder of water, and I put in another object and I displace the water, then you must know how to minus away to find the volume of an object. Then there should be some basic skills you should know. Okay, lastly, the syringe, like I said, there are two main kind of syringe that you probably will use. The bigger one would be the 10 cm cube syringe, which is able to suck up to 10 cm cube. Okay, note that on the syringe it says ml, but I would prefer if you write cm cube. Okay, these are just scientific stuff that some scientists decided that it should be written as 10 cm cube. So we just follow the routine. Anyway, this one, please don't suck any more than the 10, even though it says 11 and 12. But if not, the syringe may pop out and then everything will explode out. Okay, so the pressure will cause it to burst. Okay, also don't go and play with it. Put your finger there and try to suck your finger. Okay, just leave it be, because all the syringes are we might want to reuse it in the future. Okay, another version is maybe a smaller version. It could be a 1 cm cube syringe. That one is a bit more accurate to measure small volume. So maybe you just need 0.1 cm³ of the syringe, of the liquid, I mean, and then you should use a smaller syringe for that. One thing about the syringe, remember, do not contaminate, cross-contaminate liquids. Okay, so for example, I have two bottles A and B. I don't suck up A, then I put, I squirt it out somewhere, and then I suck out B from using the same syringe. Okay, I must always dedicate one syringe is for one bottle, and another syringe is for another bottle. And if ever I'm forced to change, I need to wash and dry. Okay, wash it, dry it, make sure there's no liquid on it, because the additional water might change the concentration, right, of the bottle. And so once it's dry, then you can reuse it again. But try to keep one syringe for different things that you're doing. Okay, anyway, here, let's talk about the smallest division of the syringe. Okay, so in this case, you will see 5 small lines, right, between 1 and 2, which means that the smallest division is 0.2 cm³. But you can still get the precision as 0.1 cm³ because you can read in between those lines. Okay. Alright, so those are all the different instruments. There may be some others, right, that we will use, but we'll get there when we get there. Okay, so this one again, I would let you see it for yourself. Just pause the video, just read it yourself. This is a bit more physics-ish. Oh, by the way, you must bring a calculator for any exam. Bio, physics, chemistry, you must bring. Just don't say bio like only write only. Bio sometimes got a calculation also. Sometimes. Okay, let's talk about the data that we collect. Okay, so do you still remember your experiment, the things like hypothesis, your constant variables, your independent variable, dependent variable. Okay, these are the things that you should still remember from your Sec 1 or Sec 2 science. Okay, alright. Now let's talk about these two things, the data that can be received from or can be obtained from an experiment. There's two types, qualitative and quantitative. Let's talk about qualitative first. Qualitative data is a quality. So quality means like green or blue, or there's a quality about it. It's just a description. Okay, a description of something. For example, a description of the behavior of a plant. The plant bends towards the light source. Okay, so if you notice your plants, right, they always turn towards the sun or they turn towards a light source. Okay, so that's just a behavior, but you didn't measure anything, right? So you didn't measure how far it moves apart. That's just an observation, a description. So it's called qualitative. Then if it's a change in color, such as a change in the Benedict's test, it's also qualitative, right? It's just orange or brick red or green. Okay, so there is a bit of quantitative to that, but you may need what you call as a color metric chart to actually see that color is equal to how much, or do that kind of thing. Okay, if you only can see your eyes are not sharp enough. Everybody got different opinion of what red means, right? Somebody's red is a bit different from another person's red. Okay, or orange is a bit different from another person's orange. So that's why it's a qualitative and not quantitative yet. Although it can be converted into quantitative if you use sufficient tools to do it. Okay, then lastly, this one, yeah, texture, right? Potato strip in distilled water. Okay, rough and hard. No, not rough and hard. Okay, it is called turgid. Turgid and hard, not rough and hard. So the plant you put in distilled water, right? Osmosis happen, right? There's a net movement of water molecules into the potato cells, the cell sac of the potato, and then right, the potato would become turgid, and then it will expand. Okay, or of course, and you must say the things like through the Paschier membrane, by osmosis, by the process of osmosis, that kind of stuff. Okay, so yeah, making good observations, right? Using your 5 senses, sight, smell, taste, touch, hearing, you need to be very detailed. Okay, I know this will be a bit annoying to write, okay, but you just have to push through and just try and do it properly. Okay, do it one time, good one, then you don't have to keep on redoing and relearning and relearning again. Okay, so let's look at some poor observations and then some good observations. Firstly, what is a poor observation? Is an incomplete description, such as the solution turned orange red. I say, hey, quite good. I already write the word turn, so good. But no, you forgot what was it originally supposed to be. It's supposed to be like that. The blue solution turned from green to orange to red. Okay, so if you actually saw it turn green, then just write it down. Okay, this is what you call as a Benedict test. A Benedict test is a test for reducing sugar. Okay, you may not have learned it right now, but you will learn it soon. Okay, and so these different things mean different amount of reducing sugar. Green means traces, orange means moderate, and then red or more red would be a large amount of the reducing sugar. Okay, so you must start from what color it was before, which is blue solution, and then turn to red. Okay, in the case of our Benedict test, you also have to write the word precipitate behind later. Okay, then let's talk about this. Okay, let's say you are doing some tests for starch using iodine tests, right? And then you use a leaf, there's some starch in the leaf, you want to test whether there is starch in the leaf. So if you write like that, some parts of the leaf turn blue black. Okay, it's a bit vague. Okay, you did not identify which part of the leaf experiences the changes. Okay, so have you ever seen some leaves, right? There is some part is white, some part is green. Okay, just show you quickly. Variegated leaf. Okay, so it looks like that. Some of the leaf is white, some of it is green. So in this case, only the green part of the leaf can do photosynthesis because it has the chlorophyll. The white part don't have. Okay, so in that kind of situation, you must say the parts of the leaf that were originally green will stain blue-black, while the parts that were originally white will stain brown after the iodine test. That means what? That means the white part has no starch, and then the green part of the leaf has starch. Okay. Lastly, if you write no change, okay, no change is not a good answer. It's a vague description. Because why? The examiner may not know what solution you are using. So you cannot say no change. He may not know that you're using iodine. He thought you're using some other potassium solution or whatever. Okay, so you have to say iodine solution remains brown. Okay, even if the examiner is the guy who set the exam, treat him like someone who doesn't know anything, right? As long as you are doing bio or any other sciences, treat the people who mark your paper like they don't know anything. Okay, don't assume that I mark your paper, I know bio, then I can mark everything for you, sweet. You just treat me like I'm dumb, and then you write all the wording out for me. Okay, now let's talk about a very important thing, biological drawing. Okay, of course, if you need to pause my video and take a break, just pause and then continue later. Okay, let's talk about biological drawings. Biological drawings are also an accurate record of your observations. There are generally 3 types of biological drawings as shown in table 6. First, drawing a whole specimen. For example, if you a leaf to draw, or I give you an orange or lemon or lime or some kind of fruit, right? Or the cross section of it, which means what? You must cut the fruit into half. Okay, so these are usually what I call as whole specimen drawing. So usually this one is quite easy to see because it's big, right? Big slices, then you can just just see it and draw it. Okay, so that's whole specimen. Second one is low power objective lens, which means you are using your microscope. Okay, this is a bit more rare. So like I told you, usually there's no budget to buy everybody a microscope each. But sometimes maybe for our own class thing, maybe it comes out. Okay, maybe. Okay, I don't want to spoil too much, but anyway, just be aware of these 3 things. Okay. Anyway, so what are the specimen under low power microscope? Will be your stem, roots, your leaf, and the distribution of tissues. Okay, so for example, you might look at your onion epidermal cells or the onion epidermal tissue, or you can even look at maybe red blood cells, red blood tissue, etc. etc. Okay, sorry, I jumped the gun. Okay, drawing of a few cells under high power objective lens. Okay, so the highest one will be at 400 times. You can draw your cells, palisade mesophyll cells, red blood cells, white blood cells, epithelial cells. Epithelial cells are like your skin cells or the plant's skin, so-called skin cells. Okay, now this is a very important table here. Please highlight, put a star, put a sticker, something on it. Okay, because these are what is commonly being tested. When you do a drawing, you must make sure you hit these 7 categories. Okay, 7 categories here. One my finger, 7. Firstly, your lines. Your lines means what? Your pencil. Now, I would suggest that everybody get one 2B pencil, the kind that you sharpen yourself, and the mechanical pencil. Okay, they have different purpose for different situation. And of course, you need a ruler. You may even want to bring a protractor sometimes. Okay, not protractor, sorry, compass. The kind that spin one. Okay, to make a perfect circle. Okay, but if you don't bring it, don't beat yourself up lah. You wouldn't die one. Okay. Anyway, please choose a hard pencil. Make sure that it's sharpened. If you're using mechanical pencil, please choose the lead that is darker. Don't choose the lead that is very, very faint. Then nobody can see anything one. Okay, so you need to draw a clear, sharp outline. Has faint outlines will make it difficult for your drawing to be assessed. Okay, basically, if your examiner If the examiner feels that he can't see it, he can't see your drawing, he's not gonna mark. Okay, he's just gonna ignore it. Okay, trust me, they are quite harsh on in that sense. Anyway, also avoid fuzzy or broken lines in your drawing. Okay, so don't like that do that kind of like sketchy thing. No, this is not art class. Just draw straight line, dark everything, or smooth and clean. Okay, accuracy. Show all relevant features in your drawing. That means that if your drawing, your whatever fruit or what got some kind of thing inside, you saw piece of seed, or you saw whatever, maybe the skin there, whatever you see, you try and draw. Okay, most of the time the question will tell you if you don't need to draw certain things. Okay, so like for example, they say, oh, don't need, you just draw the nucleus and something. Okay, then you focus on those. Okay, but if you never specify, you just draw what you see cleanly and nicely. Okay, then make sure your proportion is proportionate to one another. So for example, your nucleus is the size of a nucleus, your chloroplast is the size of chloroplast, etc. Okay, the size is very important. The size of the drawing should take up that space at least 2/3. 2/3 is 66%, but I kiasu a bit. I always ask you all to round up, just make it 70%. Okay, so fill out about 70%. Now sometimes in the exam you're given a box. Okay, so you just count, you see yourself at 70%. If they never give you a box, you just see where the end of the number is. So usually there's a mark, like a 3 marks or 4 marks kind of thing there, right, at the corner. Then you see there's the bottom. Then you just visually make a box for yourself. Like imagine there's a box there, and then you draw the size big. Okay, scared, just draw big. Okay, don't know what to do, just draw big. Okay, draw big better than draw small. Okay, next, please put a title. Okay, this may or may not get marks sometimes, but it's just good to have. Okay, add a title for the specimen that you're drawing. For you're drawing human cheek cells, write human cheek cells. If you're drawing plant epidermis, right, plant epidermis cells, etc. Then your labels. Your labels must be straight labeling line using a ruler, and ideally on one side. Okay, this is ideal lah. Sometimes you have to draw it around or both sides, but try not to cross, like the line cross each other. That'll be bad. The label line must not cross the sections of your drawing or crisscross each other. Okay, so if like let's say your cell, the cytoplasm, right, the large cell central permanent vacuole for the plant cell is in the middle, just put at the bottom, at the side, and just draw out. Okay, don't like go through the whole plant cell and cut the whole plant cell into half like that. That'll be not good. Okay, label all the relevant features of your drawing. The labels should be written away from the actual drawing so that they do not obscure any details. Okay, now you might be wondering sometimes, I draw so big already, got no space to put my labels. Okay, so that's why I said 70%, right? Then what happens to the other 30%? You use that to fill up your labels. Okay, you may need to plan a little bit, so visualize it in your brain first. Like if you imagine you draw everything, then you got space on where to put your labels, etc. Okay, so before you commit to drawing, plan first. Lastly, magnification, important. At least this one will always be 2 marks, worth at least 2 marks for your exam. Magnification is the size of your drawing divided by the size of the actual specimen. Okay, so your drawing is on paper, right? So you take a ruler, right? You measure your drawing. From which side? Usually they'll ask you to measure the longer side. So if let's say it's like a oblong shape, usually then you measure the longer side, right? From maybe the top of this part here all the way down, long one. Okay, if it's like that. If it's like that, then the same thing lah. Measure from the top to the bottom, the longest one. Unless they tell you to measure from the sides, but usually they ask you to measure the long one. Then what should you do next? You divide it by the size of the actual specimen. Okay, so how you do that? You take your specimen, maybe this is the fruit or whatever. You take a ruler, same thing, measure from the longest edge to the longest edge. Okay, measure, make sure you read it properly, and then you just do a simple division. And what must you remember for magnification? You must have this times symbol there. Okay, so let's say for example, my drawing is 10 cm and my specimen is only 2 cm. Don't need to write the cm anymore. You just 10 cm divided by 2, cancel the cm away. 10 divided by 2, 5. You put the x before the 5. So it'll be x5 sum. Okay, let's say if it's as decimal place, then express your magnification to only 1 decimal place, not 2 decimal place, not 3, just 1 decimal place. And remember the times, okay? Don't forget. It's not a lot of people always forget a minus mark because of that. Remember to put the times before you write the number. Okay, let's look at a good example of, bad example of the sketching, then a good example. Okay, so this, as you can see, very faint. Faint, not good. Already minus 1 mark already. Okay, so this, by the way, the marks usually come like that. 1 mark here, 1 mark here, 1 mark here, 1 mark here, maybe 1 mark here, then 2 marks here. Title is good, but rarely gets marked. But just still right also. Okay, let's talk about this. But acceptable biological drawing. Okay, so this too faint out, bad, minus 1 mark. Crossing of line, so your labels, it cross each other, and this one, this particular is wavy, like you own self, your hand shaky and you draw outline. No, please use a ruler. Okay, and it says this line cuts across a large portion of the drawing. So why like that? You know, can go from this side to this side, why so weird one? Okay, so not good also. Try not to cut through any important objects like this. Then shading, okay, this is not, you know, art class or what, so don't do shading. And then you see here, this one, you may say, hey, so small, only just one small little gap. Okay, small gap will kill you. So make sure that your gaps there, you really check carefully after you finish drawing. Just check, make sure everything is no gap. Okay, why? Because in real life the plant got no gap, right? Unless you tell me you go and cut with the plant with the scissors. Okay, so there shouldn't be a gap in the drawing also. And furthermore, no parallel lines were indicated where measurements were made. Magnification is not given, title is missing. Okay, so all this is all minus mark already. So what does it mean by no parallel line? It's over here lah. This is the parallel line over here. Okay, so now let's look at the example of a good drawing. Good drawing, firstly use a dark pencil. Okay, so you see dark pencil. Okay, I'm not expecting you to know all these words over here. Okay, we will get there when we get there. But for at least this is an example lah. They're just telling you must look at something like that. So all your labels all straight, you know, nicely even balanced, right? They look quite neat, and they are all tend to be on one side. Okay, of course, at some times you may need to put one on the other side, but it's fine. Okay, as long as it's neat. Then you got these so-called parallel lines that are drawing, that are indicating where you have measured. So if you started from the top and you ended at the bottom, you dot it out, draw a line, indicate the number 6.9 cm. Okay, then you see all of these objects, right? All of these features, I mean, they're all proportional to each other. They look like how it was in the plant. Okay, and then there's no shading. It's actually what? Okay, now all of this, I mean, it takes practice. Okay, so it takes a bit of practice, a bit of patience also as you're drawing. If you find that you can't do it well, just copy this diagram like 10 times. Okay, copy this exact diagram from your book until you are very good at copying this diagram. Then you will roughly know what it means to draw a good biological drawing. Okay, let's talk about magnification. So actual size of flower is 5.8, varies from the flower. You need to actually use a ruler, measure the actual flower. So 5.8, maybe somewhere like that. Okay, then you take the size of your drawing, make sure it's the same units. Don't, from one is cm, the other one is mm. Make sure it's the same unit. Okay, then size of the drawing divided by size of actual specimen will be 6.9 divided by 5.8, round off to 1 decimal place. Sorry, I messed up just now. The x you put on the right hand side after the number, not on the left hand side. So it's 1.2 times, not times 1.2. I got a bit confused because of my math. Okay, anyway, so yeah, put the times symbol on the right of the number. Okay, must remember to put this times symbol over here. Okay, let's pause here. Okay, because it's quite long. I will continue this part of the video I'll continue this part later. Okay, see you soon.